As the second week of embryonic development begins, the embryo has implanted in the uterine lining. Now, it secretes enzymes to dissolve its way into the uterine lining. And this may cause a little bit of blood to be lost. Obviously, this is a blood-rich tissue. Now, this potentially has implications in that, let's just presume that if a woman is pregnant, she would like to know that she is pregnant. Maybe she might change her diet. Maybe she's taking a medication that she would stop taking. Maybe there's some aspect of her lifestyle that she would change. Well, here we are 8 days after ovulation, so maybe around day 22 in a menstrual cycle. And as this dissolves its way into a blood-rich tissue, a little blood might be lost. And so a woman who is now pregnant might actually see blood passing through her vagina, which then she interprets as an early but weak menstrual flow. So she might feel that she has just seen evidence that she is not pregnant, even though that she is. And it is common enough that some degree of menstruation might occur for a few months into a pregnancy or even throughout. Now, one might say, well, how could that happen? Because wouldn't that shed the uterine lining where the embryo is attached? There must be some mechanism where a portion of the uterine lining can be shed, but not all of it, not an area where a placenta has formed, because it is common enough that a woman, even though pregnant, might have some menstrual flow for several months or even throughout much of her pregnancy. But here, what happens is the embryo will then have 2 layers of this trophoblast. So here, notice that all of the purple cells are separate. That's what forms the cytotrophoblast. Each cell is individual. But as these cells divide, they also then form a syncytium. A syncytium is, this happens in some places in the body, where you have a fused mass of cells. While cells divide, but after division, they don't separate into individual units. So you have this mass that has multiple cells in it, multiple nuclei, but you don't see where cells are individual. So there's a portion of this which is called the syncytotrophoblast, which forms as well. And then enzymes are produced, which then allow this embryo to burrow into the endometrium, and the endometrial lining will then grow over it so that it is then surrounded. The cells of the syncytotrophoblast then release a number of signals, the first one being HCG, human chorionic gonadotropin. It's called chorionic because these cells of the trophoblast will be renamed in the upcoming videos to the chorion. And so there is a hormone released called human chorionic gonadotropin. And this is the signal of the embryo to the mother that it exists. So this embryo would like the mother not to shed the the endometrial lining, and certainly not all of it, because that is where the embryo is attached. And so this embryo wants to inform the mother's body that it needs to shift from the original plan of menstruate because fertilization has not occurred into the alternate plan of halt menstruation during pregnancy. And so the embryo has to inform the mother's body that it exists, and it does so by making a hormone HCG. This is the first signal of pregnancy. This is what both blood and urine pregnancy tests search for. The blood tests are more sensitive, so a urine test might come up negative. A blood test then is done and it comes out positive, and say a day later the urine test is positive. But they're both searching for HCG. HCG has a number of effects on the woman's body, including causing morning sickness. Now, it certainly sounds odd that one of the first effects of the embryo will be to make its mother sick, but the purpose of that seems to be, and there is a purpose. So even though when we have a fever that makes us feel miserable and we want to take a medication to make the fever go away, nevertheless, if we make the fever go away, we might actually be sick longer. So as miserable as we feel, the fever is serving a purpose. Morning sickness seems to serve a purpose. So just in general, there are things this embryo has to do that an adult doesn't. The embryo at some point will have to, for example, decide where to put arms, where to put a nervous system, how to make a spinal cord. Adults don't have to do that. And so it turns out that in the chemicals which we ingest, many of them are nasty. Coffee has carcinogens. Broccoli has carcinogens. When we cook meat, especially the charred portions, has carcinogens. There are nasty components in the food we eat. The plants we eat are trying to kill us because we animals, we're eating them. They don't like that. And so we eat lots of nasty chemicals, but A, our liver takes care of a lot of them, so we don't have to worry about but B, certainly we don't ingest these in large enough quantities that we have to worry about. But the situation might be different for embryos. So there might be something that an adult ingests that doesn't damage the adult's body, but nevertheless is a threat to the embryo. So the drug thalidomide, it had a lot of good effects on adults, but it caused the birth defect of having, affecting limb development in infants where hands and feet would essentially be attached at the girdle. And so this was a famous case of here's a drug which was okay for adults, but which harmed fetuses. And so the challenge then is, are there things that an adult might ingest which might interrupt embryonic development when it's first forming its organs at its most delicate stage? And so it seems that morning sickness serves that goal, so that a woman with morning sickness would probably eat a little less, eat blander foods, not spicy foods. And in essence, then the embryo is encouraging the mother to avoid the sources of chemicals which might cause damage. Women who don't undergo morning sickness have a slightly higher rate of miscarriage than women who do. So once again, just like a fever, there seems to be some role of morning sickness. So once again, all of the cells of the embryo's body will come from this region. The trophoblast, both the purple cytotrophoblast and the green syncytiotrophoblast, they will not form parts of the embryo's body. This portion of the embryo's body then splits to make a hyphoblast and an epiblast. We'll get back to those in a second. But in the second week, you might think that the embryo's main priority is to get busy with making a body. But that's actually not the case. The main developments of the second week are not here, actually, per se, the future body, but rather what are called extra-embryonic membranes. So if you were to look at this embryo and count the cells here versus count all the cells here, most of the cells are not part of the body. Most of the embryo cells are actually membranes around the body, which we'll call the chorion. And then other extra-embryonic membranes form. From the region of the hypoblast, another extra-embryonic membrane called the yolk sac will form. And from the area of the epiblast, another extra-embryonic membrane will form, and known as the amnion. And so one of the main developments in the second week of embryonic development is that a large number of embryonic cells then become dedicated to these extra-embryonic membranes, which don't form part of the embryo's body per se, but rather membranes around the embryo's body. Now, another one will develop shortly afterwards called the allantois. In egg-laying animals, all of these have roles. Forgive me, I think I put that video at the end. And so in egg-laying animals, the yolk sac is where food is stored. The allantois is attached to the bladder and helps to store embryonic waste. So here's a fetal cat. And notice there was an amnion around it, a membrane. And so all all vertebrate embryos have a yolk sac, and all amniote embryos then have an amnion, a chorion, and an allantois, these extra-embryonic membranes. Now, the extra-embryonic membranes for amniotes were important because this turtle is doing something that amphibians can. It's laying its eggs on land. And so while the yolk sac was the earliest extra-embryonic membrane for food, these others help reproduction on land and were important for then the amniotes, the reptiles, birds, and mammals. However, while all of these form, even in humans, so here's a human embryo, notice there's a yolk sac, there's an allantois. But animals which don't lay eggs, like this egg-laying lizard here, they don't need a yolk sac because technically our food isn't coming from the yolk in the egg, but rather from the placenta and our mother's bloodstream. And so while the yolk sac forms, it, a few interesting things happen there, but it will ultimately degenerate and certainly not serve its ancestral role as being the food source. An allantois will form. Its blood vessels do become part of the placenta, but it doesn't collect urinary waste the way that it did ancestrally. So in this egg-laying lizard, the lizard embryo doesn't want to urinate over itself during development. So your nitrogenous waste can be collected in this allantois. And so notice that, whether it be this dog, whether it be this live-bearing lizard, whether it be this marsupial, this horse, this early primate, this mouse, that even though the yolk sac doesn't serve the role that it once did, it isn't as important, it still forms even in humans. The allantois doesn't serve the role that it once did, but it still forms even in humans. Two of the extra-embryonic membranes are very important. The amnion surrounds the embryo with a fluid, an amniotic fluid. And so we are called amniotes, we reptiles, birds, and mammals. Because we can reproduce on land, our young don't have to be laid in water, produced in water, because they do develop in their own water, in a sense, their amniotic fluid. So we don't need a pond for that. And so coming from the epiblast, the amnion surrounds the embryo in fluid. And this is important for temperature regulation. This helps to absorb shock. So that if a pregnant woman were to fall, the amniotic fluid would help absorb shock and protect the embryo. So of the four extra-embryonic membranes, the yolk sac and the allantois, they aren't as important, but the amnion is very important. And perhaps the most important then is the chorion that surrounds the entire embryo. The chorion in egg-laying animals, this is what performs gas exchange between the outside world through the eggshell and the embryo. In humans and other amniotes, the chorion still performs gas exchange, but in mammals as part of a placenta. Now let me describe the placenta. The placenta is an organ which is made of tissues from two separate individuals. The functional layer of the endometrium from the mother forms part of the placenta, and the chorion of the embryo forms part of the placenta. So once again, the placenta is a tissue. It is an organ produced from the tissues from two separate individuals. So here we're beginning at looking at the functional layer of the endometrium. So this is the maternal tissue. Now, where it touches the chorion of the embryo, there's going to be some spaces, intervillous spaces, where blood oozes out. All right, and so here once again is the human embryo, the yolk sac and the allantois. They don't amount to what they did ancestrally. The embryo is surrounded by an amnion, which produces an amniotic fluid in blue. But in red, the chorion will form the fetal part of the placenta. And it will form these finger-like projections into the endometrium known as chorionic villi, what you can see here to the right. Right, and so The chorion is the fetal portion. Once again, the maternal blood is oozing out over these finger-like projections, these chorionic villi. And so it is the fetal blood, the maternal blood, which is the source of the oxygen, the glucose, and the nutrients, etc. Let me follow that up in this next video on the placenta. The important thing with the placenta is that blood does not touch. The fetal blood, and so here is the chorion of the fetus, the fetal blood does not contact the maternal blood, which is oozing over it. So the endometrium is very rich in blood. As the chorionic villi form, they form a space between the fetal cells and the maternal cells, an intervillous space, and the maternal blood oozes over the chorion. They don't touch. So fetal blood does not mix with maternal blood. If it did, the mother's immune system would recognize the fetus as foreign and destroy it. And so this blood cannot interact. But nevertheless, because this maternal blood is so close to the fetal blood, diffusion back and forth can occur. So oxygen can diffuse from the maternal blood into the fetal blood. Carbon dioxide can diffuse from the fetal blood into the maternal blood. The mother's last meal, so glucose, amino acids, ions, can diffuse across the placenta. When the embryo makes a nitrogenous waste, urea, this urea doesn't have to be stored in an allantois. It can then cross the placenta. Unfortunately, many things can cross the placenta, including drugs, alcohol, some viruses. And so, what a woman is exposed to and, the chemicals she ingests obviously have to be, have to be controlled to protect the fetus to the greatest extent. And so the placenta is where the maternal blood oozes over the embryo, and this is where the exchanges occur. But note that blood does not cross the placenta. So week 2 of the embryo's life, its most significant aspect is that these extra-embryonic membranes are developing. As week 3 begins, a process known as gastrulation occurs. In gastrulation, cells begin to move, so there's migration of cells throughout the embryo. And differentiation occurs so that the first tissues occur. Right, so the embryo has been a mass of cells up until this point, but now it starts to form tissues. Now, these aren't the tissues which we find in adults. Adults have epithelia, connective tissue, muscle, and neural tissue. There are 3 embryonic tissues from which the adult tissues will form. The cells which line the yolk sac will form the endoderm. The cells which line the amnion will form the ectoderm. And then afterwards, in between the two, a third layer called mesoderm form. So this is the mass of cells which will become the embryo's body in the future. But in the 3rd week of life, in gastrulation, tissues form, including endoderm, consisting of endoderm, ectoderm, and mesoderm. Endoderm then contributes many cell types of the adult. And so if you consider the epithelia of the digestive tract, the gallbladder, the liver, the epithelia of the thyroid, the parathyroid, the pancreas, the thymus, the epithelia of the bladder and urethra, vagina, and the reproductive glands, they arose from these yellow cells from endoderm. So once cells have decided to be endoderm, they are choosing a cell fate. They haven't decided whether they want to be pancreas or the epithelia of the respiratory tract yet, but they know that they're going to form one of these cell types as opposed to others. The blue cells of ectoderm, they then have begun to choose a cell fate. Now, this will include the epithelia, including that of the outer skin. It includes the nervous system, hair, nails, mammary glands, the structures of the eye and ear, many glands, the pineal, the pituitary, the adrenal medulla, etc. So the blue cells are starting to choose a cell fate. They don't know necessarily whether they're going to form skin or nervous system at this point, but they do know that they're forming one of these structures as opposed to one of these in yellow. From mesoderm, this is where we're getting most of our muscle, bone, cartilage, where the blood marrow, lymphatic vessels, dermis, the middle ear, vascular tunic of the eye. So in the third week of life, the big process is that of gastrulation, where the 3 embryonic tissues form of ectoderm, endoderm, and mesoderm. As we then go into the 4th week, now the embryo begins to truly decide how to build a body. So let me just, all of these videos go into these in greater detail. First, or one of the early things that forms is a notochord, a solid rod along the longitudinal axis of the embryo. Now you might be thinking, wait, notochord, that's this primitive structure that early chordates had, that primitive fish still have today. I don't have a notochord. Well, as an adult, you don't, but as an embryo, you did. You formed a notochord from head to tail. So if you were to look at this pig embryo, you can see the notochord here. And one of the things the notochord does is it sends out signals, all right? And so that's actually an important concept here, because the cells of the embryo, they have to start forming brain and bone and eye and liver, etc. But cells have to know where they are. They have to know positional information, because the arm only forms in a specific area. The brain only forms in a specific area. So this notochord, once it forms, it sends out signaling information, which then helps the nervous system form by telling cells where they are in the embryo. Now, while we don't retain a notochord as adults, a continuous rod from head to tail, nevertheless, the intervertebral discs and the inner pulp of the intervertebral disc between vertebrae, that still contains remnants of this embryonic notochord. And so one of the things that's important, and that was one example, there are others, but that starts getting into more of the genetic angle, are signaling areas which send out signals that cells use for positional information to help them decide what genes they turn on. So here we are in week 3. The embryo is forming this more of a flattened disc. And one of the things that it does as the notochord starts to form is it decides here's going to be where the mouth is, the oropharyngeal membrane, and then here's the opposite end forming a cloacal membrane. In more primitive animals like fish, amphibians, and reptiles, they have a cloaca, which is a common opening for the digestive, urinary, and reproductive tracts. They don't have a separate, say, anus, vagina, and urethra. They all empty into the cloaca. And so the embryo on day 15 has essentially said, here's my head end, and here's my tail end. The notochord is forming deep to this. And with the signaling of the notochord, the embryo then decides this is now where the nervous system will form. So between these two, over the notochord, a primitive streak forms, and this will be the site of the formation of the nervous system. So if there's the notochord, here's the primitive streak. This will form the brain and spinal cord here. Now notice there are cells which are moving. Gastrulation is a time of, the cells are migrating and moving to different portions of the embryonic body, and thus positional information is important. If you're moving to a specific site, you need to kind of know where these sites are. We'll get back to the nervous system in a second, but I just wanted to define somites. One of the ways that you can make a body is essentially to copy and paste, to essentially, say, let's take this segment and then repeat it, and then repeat it again, and then repeat it again and again and again. And so having repeating elements, this is a way that many organisms make a body. And certainly we do, because if you look early in embryonic development, you see what are known as somites, that along the length of the body, there start to be these connective tissue blocks that repeat, going from a head to a very prominent tail. So in human embryos, you see the somites form, in a chain. Here you see a pig embryo where you see the definite somites there. And so these will be blocks of connective tissue. Here you see a chick embryo. Notice the somites forming there. We form more than we need because we form a prominent tail for a bit. Now, these will then be modified, but there are parts of the adult body which are still segmented like that. I mean, we have ribs which segment, and between them there are intercostal muscles and nerves which come out from the spinal cord. The rectus abdominis in the abdomen, it's an abdominal muscle. It could just be one single one, but it's separated into segments. Some of this segmentation is modified. Early in the hindbrain, there are segments that one can observe that are then modified. So somites are forming, and that will help guide the development of the connective tissue of the embryo. And then getting back to the nervous system begins as a flat plate, a neural plate along that primitive discs. But then it starts to form these folds, all right? So this area changes shape, it starts to form folds, and then the ends of the folds then touch each other to form a tube. So this tube is now deep, so that the ectoderm started off as neuroectoderm that could form either skin or nervous system, but now the nervous system component is deep, and what will then become the skin is superficial. So this ectoderm has chosen fates, where some will become skin only, some will become nervous system only. Here in a frog, you see here is the flat neural plate. Here you see it forming these two folds, and then the folds touch and then form this neural tube. Now notice that the neural tube was hollow. That's different from most invertebrates, where the nervous system is a solid rod, but we vertebrates, we have a hollow nerve tube. And here you can see a chick brain, a pig brain, and then a pig spinal cord. Notice that it is hollow in embryonic formation. But it still has spaces as adults. So if you study the brain, you know that there's lateral ventricles, a third ventricle, a cerebral aqueduct, and a fourth ventricle. If you study the spinal cord, you know there's a central canal that still runs through the center of the spinal cord. So our nervous system develops as hollow, and then it remains hollow throughout life. As those folds formed the neural tube, just a quick point, there were cells which were right on the crest of these neural folds that did not form part of the neural tube, but then are what are called neural crest cells. So only vertebrates have neural crest cells, and neural crest cells then produce many things that only vertebrates have. Some features of the head and face, features of the ear, the dorsal root ganglia on either side of the spinal cord. So neural crest cells are important embryonic cells that form on the crest of that neural tube. Obviously, all of the organs then go through a fascinating process of formation. I'll just quickly run through a couple. So the brain starts off as three separate regions: the forebrain, midbrain, and hindbrain. And then from this, the forebrain breaks into the cerebrum and diencephalon. The hindbrain then forms the pons, cerebellum, and medulla. And what started off as kind of just a straight region now begins to fold back upon itself, and then starts to have more of the typical human human form. If we were to go through a few other organ systems. Okay, we'll do this one then. One of the things that I'll cover in a couple of videos is the male and the female reproductive systems. They start off the same, but there are ducts which only the males will keep. So males will keep the blue Wolffian duct, but lose the pink Mullerian duct. The blue Wolffian duct will form part of the vas deferens, whereas the pink Mullerian duct forms the oviduct and part of the uterus. And so male and female embryos, they look identical for early development, and then they start to modify in one gender-specific form or the other. Now notice that the kidneys started actually inferior to that. The formation of the kidneys is actually much more complex than most people realize. In that fish kidneys stretch the entire length of the body just on either side of the vertebral column. And so fish kidneys are long and thin. We could break them into regions: the pronephros, the mesonephros, and the metanephros. Now, as evolution occurred, getting into the amniotes, what ultimately happened was the pronephros then became non-functional. The mesonephros was lost, although some of this is kept as the vas deferens and that Wolffian duct. And this bottom portion, the metanephros, ends up being what becomes the adult kidney. But as embryos, we still form all of these. So in embryos, kidneys stretch the entire length of the back, but then most of them degenerate. But then these metanephric kidneys, the ones which will be kept, they are in the pelvic region, and then they then have to raise. And so much of the embryonic kidney then has to be broken down, and the metanephric kidneys, they then have to then rise, and sometimes there are birth defects associated with an abnormal movement of those. I'm sorry, I didn't mean to do that. The human embryo also possesses a cloaca, and the cloaca then must subdivide. So here you see from the previous video, the kidneys must go up, the gonads must go down, the gonads form high in the body. But then this cloaca, which was one original opening, then gets separated into an anus, a urethra, and in females, a vagina. So in, say, fish and amphibians, these are all empty into one solid opening, but the cloaca then needs to divide. And in humans, the cloaca does form, but then must be subdivided. Just a couple more quick points. There are cells that have to migrate from one spot of the embryo to another. One of the interesting things in the reproductive system is the cells of the ovaries and testes, which will produce gametes. They don't originate in the ovaries and testes. They have to find them. So they actually form close to where the yolk sac and the allantois meet, these primordial germ cells. They then have to migrate to the site of the ovaries and the testes. Many of the bones which will form are what are called composite bones. So if you consider reptiles and fossils, one doesn't find an occipital bone, a temporal bone, a sphenoid bone. Instead, one finds little pieces. So there's lots of little pieces of bone in the occipital region. So there's exooccipitals, basooccipitals, postparietals, tabulars, etc. So what we see in human embryos is, once again, an occipital bone doesn't form, but lots of little pieces that then all fuse together. So a lot of the bones that we have are actually composite bones, where they form from the fusion of multiple separate bones, which not only form as different sites in the embryo, but then also then if you look at fossils and reptiles, etc., ancestrally little bones fused together to make our big single mammalian bones, and that we see that happening in embryos as well. Okay. Before I get into the last topic of fetal circulation, just a couple of things. And so if you were, say, studying genetics or this aspect of human physiology, I mean, this is, fascinating. How does a ball of cells become human? I mean, it's an incredible question, an incredible biological topic. And much of the answer is in what genes are expressed. Very many of the genes of the embryo are different from genes of the adult. We've already mentioned that fetal hemoglobin is different from adult hemoglobin. Fetal hemoglobin uses gamma globin instead of beta globin. And so there are genes. And so one of the great genetic questions is, how does one regulate genes? So it's not just a question of what genes one has, but what genes one is using. So when do you turn a gene off? When do you turn it on? And so liver cells are expressing only liver genes. So out of all of these different genes, one cell is using only some, one cell is using only another. So certainly there's a great opportunity to study the genes of embryonic development. And if you were to consider like the globin family, you would see that here's a bunch of globin genes in the beta cluster, but at different stages of embryonic development, this one is used. Right, so the first trimester, that one is expressed. In the second and third trimesters, that one's expressed. After birth, this one's expressed. So the timing of when one uses genes is important. Before getting into the last topic, just once again, embryos are different from adults. So look here, you can see the retina. The retina actually forms as part of the brain, part of the diencephalon, and then stretches away from it. The hindbrain has segments, which the adult hindbrain does not. The limbs start as flipper-like structures. Here you once again see somites, you see the prominent tail. Sometimes human infants are born with a tail that must be removed at birth. One of the fascinating things is in, say, fish and amphibians, you can form gills. There are there are slits in the throat where gills form. Well, this is a chicken. Chicken embryos, pig embryos, and human embryos still form these pharyngeal arches, these pharyngeal slits, with the blood vessels that went there, et cetera. So we still form a throat similar to the way that organisms which will make gills form it. Now, we then have to reorganize everything, because in this embryonic throat, the thymus is up here, the thyroid's down here, they have to switch places, and every now and then there are birth defects because of abnormal migration of these tissues. We form a notochord as embryos, but some adults still have notochord persisting where it shouldn't. Some even have cancer of the notochord later in life. The heart starts off looking like a fish heart, with one single atrium, one single ventricle, and then other chambers, a sinus venosus and a truncus arteriosus. As these subdivide to make the human heart, sometimes there are abnormal divisions of these vessels, which is a problem, potentially a fatal one. Many alternate circulatory patterns form because we often begin with, say, fish blood vessels. Fish have different blood vessels than humans, and human embryos then start off making, say, cardinal veins and subcardinal veins, only to realize, at some point, oh, wait, I don't want these. I want the adult humans. And so we reorganize our circulatory system, but very often we have abnormal blood vessels resulting. Our first red blood cells have nuclei just like, say those of reptiles. When the yolk sac and the allantois forms, it herniates the intestines. The intestines are actually outside the body for a bit. Now, while they typically return, there are infants born with a herniation of the intestines that can cause great difficulty. We form a cloaca, and the cloaca then has to subdivide. But this subdivision of the cloaca, that can then be a problem, because then, the anus could empty into the vagina or the urethra in males if the cloaca does not divide normally. The kidneys, once again, form all along the back, but then the kidneys have to change position. They have to rise and the gonads have to fall, but this can then cause problems. I'm sorry. My bad. Where you can still have individuals with a pelvic kidney that does not ascend, or a horseshoe kidney, or abnormal ureters. The uterus and vagina form from a pair of tubes with fuse, but sometimes this does not occur normally, where there can be a T-shaped or a Y-shaped uterus, which is more like that which we see in other mammals other than primates and humans. Or even a partial septum in the uterus or the vagina. Mammals develop a mammary ridge from which mammary glands can develop, and thus, where mammary glands occur in dogs is different from cows, which is different from bats, and some species have it under the armpit, some it's more abdominal. Well, extra mammary glands, extra nipples, and extra mammary tissue can form in humans anywhere along this mammary ridge. And so understanding human development then also helps understand a lot of birth defects, which then have to be corrected or treated after birth. Finally, the circulation of blood during embryonic development is different from that of the adult. So a quick review. When blood leaves the left ventricle, goes into the descending aorta, it reaches the hip region, goes into iliac arteries. But in the same region, there are fetal arteries known as umbilical arteries that the adult doesn't have. So the blood passes from umbilical arteries to the navel and then leaves the body. So these umbilical arteries leave as part of the umbilical cord and then go to the placenta. So this then is the blood supply of the chorion, the fetal part of the placenta. And here gas exchange occurs with the maternal tissue. Thus now the blood is oxygenated. So it is thus in the placenta that the oxygen supply is obtained, not the lungs. The blood comes back through an umbilical vein. And the umbilical vein then once again passes through the navel and then reaches the liver. Now, the liver is super complicated. This video will go into it in greater detail because now the liver has different regions. And then there are different regions of the liver with different levels of oxygenation of the blood. So the liver is very complicated. Significantly, though, there is a branch off of this umbilical vein called the ductus venosus, which will now connect to the heart. It's kind of cone-shaped. That's important, as I'll mention in a second. And so thus oxygenated blood can pass from this umbilical vein, which came from the placenta, and then reach the heart through this ductus venosus. This is an important structure. There are some potential problems in fetal development if this ductus venosus is modified or is absent. In the fetal heart, the ductus venosus enters the heart just as the inferior vena cava does. And at this stage, the two atria merge and do not have a septum between them. So there is an opening, a foramen ovale, which connects the two atria. Now, while one might think that this would mean that all of the blue and red deoxygenated and oxygenated blood is mixed, that's not entirely true because the ductus venosus enters at a certain angle, its cone shape gives its blood a different speed, and so these two factors actually do largely separate the red oxygenated blood, which is mostly heading to the left atrium, from the blue deoxygenated blood, which is mostly heading to the right atrium. But once again, the fetal heart has a hole, an opening between the two atria. At birth, a flap should close over it and form a septum. So now red oxygenated blood reaches the fetal heart. If it should mostly go from the right atrium into the left atrium through that foramen ovale to the left ventricle to the aorta, and then reach the aorta, and then from there hit the arteries, which will go to the brain. Notice how red they are. So notice that the fetus is placing a priority on getting red oxygenated blood to the brain. But a couple of other structures should be mentioned. So some blood, it's purplish, part oxygenated, part deoxygenated, gets to the right ventricle, which then goes to the pulmonary trunk. Now, from the pulmonary trunk, some of it should go to the lungs. I mean, the lungs have cells, they need oxygen, so obviously some will go there. But you certainly don't want to send half of the blood leaving the heart to the lungs, because they will not have any oxygen in them until the moment of birth, when the infant takes its first breath. And so a structure exists that does not exist in adults, a shunt which connects the pulmonary trunk to the aorta, what's known as the ductus arteriosus. That way, most of the energy pumped by the right ventricle doesn't go to the lungs, which once again need blood, but they're not, providing oxygen, so there's no need to send half of the heart's blood supply there. The ductus arteriosus will then take this blood to the aorta. Notice that it joins the aorta only after the arteries going to the brain have separated. That way the brain is getting more oxygenated blood than most of the body. This blood then goes, it's purplish now, it goes in the descending aorta, and from there much of it can be sent to the placenta to be oxygenated again. And so thus fetal circulation is different from what we see in the adult. And so these two videos have been an attempt to highlight some of the main features of fetal development, but it is truly a wondrous process to take a bowl of cells and to convert that into a human. And so thus these videos go into this in a little more detail, and certainly any further discussion should certainly then get into many of the genetic aspects as well.